Hello, my name is Grant Kramer, and I'm a professor emeritus at the University of Nevada, Reno. Today, because of special requests for videos on mineral nutrition, I'm starting a new series on the mineral nutrition of plants and specifically grapevines. So my background is originally in mineral nutrition. Before I started back into school again, I was very interested in what was the difference between organic and chemical fertilizers while living on my uncle's farm in the middle of Massachusetts. So I went back to school. I took some classes on organic versus chemical mineral nutrition of plants and found it very interesting and very exciting. And there's a lot I've learned about that, and I'll speak about that in an upcoming video. But today I want to talk a little bit about my background of working in Emanuel Epstein's lab. As a student, I studied and I worked in his lab, which he is the premier scientist, the premier plant nutritionist. He wrote the book on the mineral nutrition of plants, and I feel very fortunate to have been there when he was in his prime and doing some very exciting research. So that's how I started, in the basics. So my part one of this series is going to be on the basics of mineral nutrition, the generalized background that you find. That will be in this video. In the second video, I'm going to give an overview on the grapevine specific nutrition. So you'll be interested in that if you're interested in grapevines. And then finally, I'm going to go into more specific detail about individual nutrients. For example, nitrogen is very important in grapevines. It impacts the vigor of the plant and the way it grows, but also impacts the quality of the grapes. So let's get to it.
The mineral nutrition of plants, part one, the basics. Here you can see me on the right-hand side in a trench. This demonstrates how the soil profile can change with depth. We'll discuss this and other factors in this coming video. To make it easier for you to get to different parts of this video, I've created a table of contents in the approximate minutes and seconds that they start. So plants have very simple requirements, very basic requirements. First of all, they need light to provide photosynthesis or energy for the plant. They need CO2 for photosynthesis and the physical body or structure of the plant. They need water for both their body structure and for biochemical reactions to occur. And they need minerals to build their body structure as well, but also for many biochemical reactions. Plants do not need vitamins as they are able to synthesize all that they need. So they need light, CO2, water, and minerals, and that's it. So plants are made up of mostly water, anywhere from 50% water in trees to 95% water in vegetables. If we look at the percent dry matter of each element in the plant, we can start here with hydrogen, carbon, and oxygen. And all of these are expressed as a percent of their total dry matter. And they're also expressed as the relative number of atoms with respect to molybdenum. Molybdenum is the element that is in the lowest amount in plants. Hydrogen has 60 million atoms relative to molybdenum and represents about 6% of the dry matter. Carbon represents about 45% of the dry matter and 40 million atoms relative to molybdenum. And oxygen represents 45% of the dry matter and 30 million atoms in respect to molybdenum. And these are obtained from carbon dioxide and water. Mineral elements are broken down into two different categories. First, we have the macronutrient elements, and in the next slide, we'll talk about the micronutrient elements. So macronutrient elements are elements that are needed in larger concentrations by the plant in order to develop and reproduce. So these elements that I am showing you are all essential elements. They're elements that are required both for the development of the plant and for it to reproduce itself. So let's start with nitrogen. which is the chemical element that is needed in largest concentrations. And it's typically found in the dry matter at about 1.5%, or about 1 million atoms relative to molybdenum. The next element is potassium, seen here with a chemical symbol of K. And its concentration is 1% in dry matter, and it has 250,000 atoms relative to molybdenum. The next element with the highest concentration is calcium, and it represents about 0.5% of the dry matter with 125,000 atoms to molybdenum. Magnesium is next at 0.2% and 80,000 atoms relative to molybdenum. Then comes phosphorus at 0.2% at 60,000 atoms relative to molybdenum, followed by sulfur at 0.1% at 30,000 atoms per molybdenum atom. And then a relatively newly recognized element is silicon. Silicon is everywhere in the environment, but it was not recognized as an essential element until fairly recently, much to the work of Emanuel Epstein, who helped to define this. So it is also in the concentration range of about 0.1% of the dry matter and 30,000 atoms with respect to molybdenum. Micronutrients, as I mentioned, are required in less concentrations. And in fact, there's a very fine balance for micronutrients, because if you're too little, you're deficient. If you're too much, then you are toxic. So micronutrients are tightly controlled by the plant in order to maintain a good balance. I should mention that most of the nutrients that we're talking about are needed in photosynthesis, but there are other specific functions in plants besides the photosynthetic apparatus that require these elements. So in terms of the micronutrients, chemical symbol for chlorine or chloride is Cl. Chloride is the anion of chlorine, which is the ionized form of it when it's soluble in water. The concentration in dry matter now is not listed as percent, but in parts per million, and there are 100 parts per million chlorine or chloride, and there are about 3,000 atoms with respect to molybdenum. Next is iron, shown here with a chemical symbol of Fe, and its concentration is 100 as well, but only 2,000 atoms relative to molybdenum. Boron, another important micronutrient that can be very deficient or toxic and have very dramatic effects on the plants, is in the concentration range of about 20 parts per million and 2,000 atoms relative to molybdenum. Then we have manganese at 50 parts per million and 1,000 atoms relative to molybdenum. Then zinc at 20 parts per million and 300 atoms relative to molybdenum. Copper at 6 parts per million and 100 atoms relative to molybdenum. And then nickel, and finally molybdenum. Okay, so let's talk about the general principles of how plants take up minerals. First of all, plants principally take them up through the roots, although it's possible for them to absorb mineral elements or nutrients through the leaves, even through the waxy cuticle if it's sufficiently provided with a surfactant to allow it to penetrate through the cuticle, and easily through the stomatal pores when they are open. So how, you might ask, would minerals get in there? Well, it would come from the gaseous air in the environment or through rainfall and the water droplets. So for example, air pollution can have sulfur in it, and that could add some sulfur to the plants and alleviate some deficiencies, for example, if the soil was deficient in the sulfur, and that's been seen before. For minerals to be taken up, they first of all have to be dissolved into ions, that is, as charged particles in the soil solution. They cannot just contact the mineral and absorb it. They must dissolve into solution, become into ionic form, and then they will be taken up by the plant roots through specific uptake mechanisms that we will not discuss here. And that amount that is taken up is concentration dependent. So in higher concentrations, more can be absorbed. And if it's in lower concentrations, then in solution now, we're not talking about in the soil, but dissolved and in solution, then it will be taken up. So just because the mineral's there doesn't mean it's absorbed. It has to get into solution first. Some minerals are mobile in the soil and can move towards the root as water is taken up by the plant. So the plant is transpiring, it's sucking water towards the roots from afar. And if those mineral ions are soluble and able to move through the soil particles, then they will move closer to the plant. And I'm going to discuss this a little bit more in detail. Others are very immobile and can't move, and the roots have to grow into the region in order to explore the soil and effectively mine that soil to get those minerals. So roots are seeking out for minerals. And roots will proliferate, in fact, in zones where there's high fertility. They'll produce more roots in that area in order to absorb those nutrients. Another important factor to think about is the mobility of the ion in the plant. And the reason for this primarily for you here is so that it helps you to understand where and why a mineral deficiency symptom shows up in a plant. So ions move in two different systems in the plant. One is the xylem system, which is the water pipe system that is absorbing water and transferring that water from the roots up to the leaves. It's in one direction primarily. And the phloem is another piping system, which is full of sugar that is produced and moved around based upon a source to sink relationship, which I don't want to explain too much here. But suffice it to say that where the sugars are produced, like in the leaves, they are sent to other parts of the plant which require those sugars to grow. So, for example, the apex of the shoot tip, or the tuber of a potato plant, or the root systems can't produce their own sugars. They're sent by the leaves, through the phloem, to those places. And a number of those elements are very mobile in the phloem. They can get into those living cells and move along with, by mass flow, with the sugars. However, certain mineral elements are not mobile. That is, they cannot be in sufficient concentration in the phloem without causing detrimental or negative effects in the plant. And so the plant reduces or doesn't allow those concentrations to occur. So let me discuss those mobile elements and then discuss the immobile elements and then explain why they're important in terms of where they show up in the plant. So first of all, mobile elements will show their deficiencies first in the older leaves because the plant can recirculate those nutrients to the newer tissues that need it. Immobile elements cannot be recirculated from the older parts of the plant to the new plants. Hence, mobile element deficiencies will show up in the older leaves first, and immobile element deficiencies will show up in the young parts, the young leaves or the apex of the plant first. So nitrogen is a mobile element and readily shows deficiency symptoms of a chlorosis, a yellowing of the leaves in the older leaves first. And it's mainly transported in the phloem as amino acids and also or nitrate, depending on the species. It varies from species to species. Potassium is a mobile element. Magnesium is a mobile element. Phosphorus is a mobile element and is transported as adenosine phosphates like ATP and also phosphate. Chlorine is transported as chloride. Sodium is transported in the plant as sodium. And molybdenum is transported in the plant as molybdate. So those are all mobile elements. Then we have a category of elements that are what we'd call partially, or what I would call partially mobile, and it depends on the species. Some are able to transport them more readily than others. Sulfur fits into this category. So as a result, sulfur has a very similar look to its chlorosis as nitrogen does. And it's a general yellowing of the leaves, but it shows up primarily in the younger leaves first and not the older leaves. But again, this varies a little bit with species, so you have to be careful about identifying mineral deficiencies, and it's species dependent in many cases. Zinc is also one that is partially mobile. And then we have the immobile elements, which are calcium, which show very significant deficiencies in the shoot apex, as well as boron, where the plant will literally die. The apex will die because of deficiency in that area. Iron is another immobile element, and thus it shows its interveinal chlorosis, a pattern that you'll see quite distinctly later in the presentation, that is showing up in the younger leaves. Magnesium, on the other hand, shows a similar interveinal chlorosis deficiency, but it shows up in the older leaves first. So this is why it's useful to be able to identify the differences in the deficiency symptoms. Manganese and copper are also immobile elements. So as I mentioned, most elements come from the soil. Soils by their nature are highly variable. First of all, structurally, texturally, they're made up of different sized particles, which are defined as soil texture. So we have sand, silt, and clay particles. Sand is any particle between 20 to 200 micrometers in diameter, and it has a very low cation exchange capacity. I'll explain what cation exchange capacity is in the next slide. Silt is between 2 and 20 micrometers, and its cation exchange capacity is moderate. And clay is any particle less than 2 micrometers, and it has a much higher cation exchange capacity than the other silt and sand particles. There are different kinds of clays that have different fertilities or cation exchange capacities, such as morillonite or kaolinite, just to make things more complicated. So cation exchange affects soil fertility and the mineral content, and that's why it's important. And it varies with the bedrock that it comes from, flooding conditions, and the weather or rainfall. Organic matter is an important component of the soil as well, and it acts as a slow-release mineral fertilizer. We'll talk about this more in future videos, but I'll talk a little bit about this in this video as well. And organic matter also contributes importantly to the water holding capacity of a soil, particularly if it's a sandy soil that drains very quickly. Organic matter is really important to helping to hold that water in the soil. It's important for soil structure and, as I mentioned, water drainage. So what are we talking about with cation exchange capacity? So each particle can have negative charges on it, and that's what you see here in the left-hand side of the slide. Those little lines are meant to be negative charges. It's a very crude, simple model here. Soils are much more complicated than this. Suffice it to say that with negative charges on the soil, cations are going to be attracted to it. They have a plus charge, and then particles in the soil are negative charges. They'll be attracted to each other and adsorb to the soil particle. They're not bound permanently. They're capable of coming on and off or away from that negative charge because ions have energy to them, and they can move on and off by either popping off by themselves or being displaced by another cation. And this is also concentration dependent. But up here on the top, see a model of that cation exchange where a magnesium ion here is displacing a calcium ion, knocking it off the complex. And therefore, it's bound to the ion, but now the calcium is free in solution. This also can happen between potassium ions themselves, so it doesn't have to be a different kind of cation. It can be one that is different. And as you can expect, with 2+ charge, there's a stronger attraction than there is with a 1+ charge. So these ions on the cation exchange complex are in equilibrium with the ions in solution. And so as the concentration of ions in solution goes up, say by adding fertilizer to the soil, then there'll be a higher incidence of those ions on the soil particles, and vice versa. As the ions are taken up out of solution by plants, then this will cause some more ions to be coming off of the negative charges on the soil particles and going into soil solution. And this is why over a long period of time, over thousands of years, soils can keep giving more nutrients to plants. They don't just use up all the ones in solution because more keep dissolving out of and off of the soil particles. So weathering and mineralization affect the fertility of soil. Weathering is the breakdown of rock into the soil by the environment. So mineral elements will vary with the parent material that was formed in the formation of the earth. As those materials weather, they start to contribute those minerals to the soil and the soil solution. Now, once they're in solution, as I mentioned, they can move with the water, and high rainfall in the tropics will leach these mineral elements out of the soil, take them to the rivers and back out to the ocean. Whereas in deserts with very little rainfall, most of those mineral elements are still present. So naturally you find places where there are higher amounts of rainfall that the soils are depleted over time. Their mineral nutrients have been removed or leached away. Whereas in deserts, where many of our productive soils are on the earth, they're relatively fertile because of the fact that they haven't been washed away over time from the rainfalls. Another process that's important about soils is a process called mineralization. And this is defined as the process of decomposition of organic matter by the microbial populations in the soil. Usually mineralization is referred to as for nitrogen, but it can apply to other nutrients as well. And there's a very specific microbial interaction in nitrogen mineralization. For example, amino acids are converted to ammonium, as you can see here in this diagram on the right of the nitrogen cycle. So we have organic nitrogen converted to ammonium, which is NH4+. It's a cation. So it has a positive charge and is attracted to the cation exchange complex in the soil. But that ammonium can also be converted into nitrate. This is also a process that is performed by microbial populations and is known as nitrification. That is the conversion of ammonium to nitrate. Nitrate is then taken up very readily by the plant. Ammonium can be taken up by the plant, but can have some toxic problems if too much of it is taken up. So this is a very important process in the soil, from mineralization, from taking organic nitrogen, that is the nitrogen that was absorbed by previous plants, and then decomposing it into ammonium and then to nitrate, where it then is retaken up into the plant. Some of this nitrogen can be lost to the air through a denitrification process or through leaching, because nitrate, having a negative charge, moves readily, easily through the soil without being bound by the cation exchange complex. I should have mentioned that I could spend 30 hours lecturing on mineral nutrition, as I taught a course in this before. So there's a lot to this that can be expanded upon, and hopefully I add a little bit of it in future presentations. So there are fertilizers that can be added to the soil to affect the fertility, as some soils do get depleted with time. And these can be inorganic fertilizers or organic fertilizers. That is, organic fertilizers would be fertilizers that are considered organic by organic growers. That's one definition, which is that they are not chemical, and we'll get to that in the future video. Or simply organic is defined by organic chemists as any material with carbon in it. So that would represent natural living organisms that are then dying in the soil and releasing that fertilizer for nutrition of the plants. And these fertilizers, of course, can improve the plant nutrient status. So the nutrient status of a plant can be identified by its visual symptoms, or we can measure the nutrient concentrations of the soil, or we can measure the nutrient concentrations of the plant. And again, I'll speak more about this in a future video. Another food for thought here that most people don't realize or think about is not only does soil become very variable, and so you can see soil mineral concentrations being different in one area of the field than another area of the field, or on one side of the plant from the other side of the plant, because the soil is different there. or the fact that nutrients do move in the soil. And so you actually have very sharp gradients close to the roots, depending on the mobility of the ions in solution. So we call this movement of soil nutrients conductivity. And it's a result of water moving, as I mentioned earlier, to the root from transpiration, uptake of water into the plant, carrying those ions with them by mass flow towards the surface of the root. Now, anions move through the soil with very high conductivity because they have negative charges. So nutrients like nitrate and chloride are going to move very readily. On the other hand, some negative charged ions don't move very well for a different reason, and that's the one important one is phosphate. Phosphate precipitates out very readily because it binds with calcium. And calcium phosphate is basically what our bones are made of. So you can think of it as a very inert material. It's very difficult to keep calcium and phosphate next to each other without them precipitating. So phosphate doesn't move very readily at all. Cations also move differently depending upon their charge, and some will move more readily in the soil than others, but not nearly as well as negatively charged particles like nitrate and chloride. So what I want to show you in this figure on the right is the nutrient concentration in the soil solution adjacent to the root surface. And remember, the concentration influences the rate of uptake. So if we take the blue line here, which is a nutrient in low nutrient concentration and low conductivity, that is, it doesn't move very readily through the root. If we get right next to the surface of the root, we see that it's almost at zero concentration. And if we move away from some distance, maybe just a few inches from the root, we can see the concentration might be up to some artificial number here of 40, okay? Which is what we might measure if we were to measure a soil solution concentration or soil concentration of that element. But in fact, it might be quite a bit lower next to the root surface. The same nutrient at higher nutrient concentration would be in this green line. And here again, you would see maybe out in the soil it would be about 100. But it gets depleted as it gets closer and closer to the surface because the plant root is taking up the ions faster than the water can deliver it. So we're actually depleting the nutrient. On the other hand, with the gray line, we have just the opposite. This nutrient is highly mobile in the soil, and it gets carried at high concentrations to the surface of the root and actually starts to accumulate to a much higher concentration than it does in the actual soil. And you could imagine this with ions like sodium and chloride, which we know as salt. And in many cases, when we have salty soils, this is in fact what is happening. The salt is actually accumulating to a higher concentration right near the surface of the root, having detrimental effects to the plant. This is a very important slide. So this slide states that the solubility and the availability of nutrients is dependent upon the soil pH. And so it's very important to adjust your soils to the proper pH. Again, if you don't have it at the proper pH, then those nutrients may not be available to the plant, even though they're in the soil. And each mineral has its own specific pH profile, as shown here on the figure on the right. The ideal pH for most plants, and again, this is for most plants, there are exceptions to the rule. There are plants that are adapted to high pH soils and plants that are adapted to acidic soils. But most plants thrive and do well between the pH of 6 and 7.3 because this provides the optimum solubility for all of the mineral elements that are needed by the plant. If you get below pH 5, then you can run into some other issues, as you'll see here in a moment. But one important one that's not shown here in this figure is that of aluminum. So aluminum is toxic to plants, but it's in high concentrations in the soil. But at a pH below 5, it becomes very soluble. At a pH above 5, it's insoluble and doesn't have an effect on the plant. So it's only at below a pH of 5 that aluminum becomes a problem. In other cases, the pH is above 7.5, then there are elements that become very insoluble and become deficient. But other elements may become higher in concentration and may actually become toxic. Finally, the other important factor that pH affects is the microbial activities in the soil. And ideally, in that range of 6 to 7.3, you have more microbial activity going on. So let's look at these nutrients for a moment, because there are some important facts here that are important to understand in regards to your soil nutrition. So nitrogen has a pretty broad range of availability, particularly above 5 and all the way up to 9 almost. Okay, so we don't worry too much about the pH effects of nitrogen. On the other hand, phosphorus, which is another major macronutrient. So nitrogen, phosphorus, and potassium are our most important macronutrients and what are components of our fertilizers that we add to the soil. Below the pH of 6, phosphorus becomes very low for available phosphorus in the soil solution. And therefore, it's very important to keep the pH higher to allow more available phosphorus to be taken up. Potassium is broadly available. Sulfur is broadly available, except at pHs below 5. Calcium is broadly available. Magnesium becomes much more available at higher pHs than it does at lower pHs. Now here's an interesting difference. We have some micronutrients that behave in the opposite way. Iron is very soluble at acidic concentrations to the point that it becomes toxic below pH 5, as well as manganese, and even boron, and copper, and zinc. So it's this optimum range that's important. And notice as we get above 7.5, the iron availability becomes very small, as well as in manganese and even boron. So this is where, if you have high pH soils, you're going to easily see iron deficiency in plants, for most plants again. Okay, so how much nutrient is enough nutrient? So scientists have defined what are called the critical mineral nutrient concentrations. And they base this upon the growth of the plant, here shown as a percentage of some sort of maximum depending on the plant. And so this nutrient is in the sufficient zone when anywhere between 90% to 100% of the growth of the plant is maximal. So in a deficient zone, the plant is going to be limited or inhibited. It will not grow very well because the nutrient is limiting. And you can have all the other nutrients you want, but if this one nutrient is limiting, it limits the growth of the plant. It limits its productivity. So then in this zone, which is known as a deficient zone, which is anything below 90% of maximum growth, the plant growth rate is very linearly dependent upon the concentration in the tissue. And then once we start adding more and more nutrients, we start to see diminishing return to the point where there's really no further advantage to keep adding fertilizer to the plant because it won't get any greater growth or productivity out of it. And at some point, we reach a zone where that concentration starts to become toxic, and we actually inhibit the growth of the plant with too much nutrient, as shown here in the toxic zone. So we have 3 zones: the deficient zone, the sufficient zone, and the toxic zone. And it's based upon the nutrient concentration at approximately 90% of the growth rate of the plant or productivity of the plant is known as the critical mineral nutrient concentration. So another interesting point is that plants can grow without soil in solution culture if there are the essential mineral nutrients that are needed for the plant to grow. So on the left, we have a system called hydroponics, where the plants roots are growing in water. Now, most plants would not grow well in water by itself because they become oxygen deficient. So in these types of systems, it's important to have an air pump with an air stone that bubbles some oxygen through the solution to grow. But there's even a better system which has more oxygen and air in the environment called aeroponics, where instead of growing them in water, you spray or make a mist that coats the roots, but the air is around the roots. And this is actually really wonderful for plant roots. It simulates what's going on in the soil, because in the soil you have little air pockets with different sized cavities, and some have more water in them and others have more air in them, and you get this sort of balance of oxygen and water. And plants will grow really well in such a system. Okay, finally, I'm just going to wrap up by showing some of the nutrient deficiency symptoms that can occur, and this is the advantage of hydroponics. You can control the concentration of the nutrients in hydroponics and then be able to identify and visualize the nutrient deficiency symptoms. And this was an experiment I did in my mineral nutrition class back in the 1980s, in Emmanuel Epstein's mineral nutrition class, where we grew tomato plants in hydroponics with a complete nutrient solution, which is listed here as the control, and then some sort of deficient amount of a particular nutrient. So in the case of nitrogen, we gave it about 10% of the concentration that was in the normal solution. We needed some in there, the plant would just sort of die. But you can see on the left here, the control plant is much larger, and we use these lines here. It was nice to photograph them this way to be able to see size. And you can see that the nitrogen deficient plant is a slightly duller green, a little bit more yellowish. And if you were to zoom in on the leaves and the older leaves on the right here, you'd see the color differences, this dark, rich green for nitrogen sufficient plant and a nitrogen deficient leaf that is an older leaf that is showing very much a yellowing color. And this is because nitrogen is part of the chlorophyll molecule, and this chlorophyll molecule breaks down, showing some yellow color rather than green color. Chlorophyll has a green color to it. Here's another plant with phosphorus deficiency. We did it the same way. You can see it had an inhibition of growth. And again, we could have inhibited the growth more by reducing the phosphorus concentration even more. So it's highly dependent upon the phosphorus concentration as to the symptoms that we see. You can also see that the leaf size is quite a bit smaller on the phosphorus deficient plant. And on the underside of these tomato leaves, there's a purplish coloring. This is a common deficiency symptom in plants with phosphorus deficiency. But again, this is highly variable depending on the plant species and even the plant cultivar. Different plant cultivars can show different deficiency symptoms. Potassium, here again, there's a stunting of the growth, but here there's also a characteristic of potassium deficiency that you see in the leaves. And this is the formation of little brown spots along the margin of the leaves, which first show up along the margins, and as the disease progresses, it will move to the interior of the leaf, and eventually the whole leaf might turn brown as a result of it. This can also be a symptom of some sort of other toxic element, so one has to be careful in the interpretation of these symptoms. And here's a couple of examples of micronutrient deficiencies. So here we see iron deficiency, with quite dramatic effects on the plant size and the yellowing of the leaves here. And this tomato plant, again, iron deficiency generally shows up in the younger leaves, and you get this beautiful interveinal chlorosis where the veins stay green, but between the veins it is yellow. And this symptom, if you just looked at the leaf, you would say, oh, that's iron deficiency, but it could also be magnesium deficiency. Magnesium deficiency looks very, very similar to this. And the way you can distinguish them is that the iron deficiency would be showing up in the younger leaves, and the older leaves would remain green, unless the plant is really severely iron deficient, as in this one, where you may start to see it progress not only from the young leaves, but all the way into the older leaves. So it's important to look at the deficiency symptom over time. The other is magnesium deficiency, and magnesium deficiency would show up in the older leaves first, and the younger leaves would stay green. So why is this? It's because of the mobility of the ions. Iron deficiency, iron is immobile in the phloem and cannot move through the plant. And magnesium is mobile, so it can redistribute, it can recycle that magnesium from the older leaves to the younger leaves where it's needed. And I just love this picture of manganese deficiency. You do not see this very common. You do see iron deficiency quite frequently. It's one of the most common deficiencies, particularly in high pH soils. But manganese deficiency is rare. You hardly ever see it. And this is just the beauty of doing such an experiment like this in this fine intervenal chlorosis pattern that's quite distinct from the iron deficiency symptom. So in summary, mineral nutrients are taken up once they have dissolved into the soil solution. Plants require mineral nutrients. 7 of those are macronutrients. That is, they're needed in higher concentrations in the plant in order to grow, develop, and reproduce. And there are 9 micronutrients, nutrients that are needed in much lower concentrations than the macronutrients. Nitrogen, phosphorus, and potassium are the most important macronutrients and are the main components of most fertilizers. Too little of these mineral nutrients causes deficiencies that result in reduced plant growth, but too much can cause toxicities as well. So a grower is striving for a balanced nutrition from rich, deep, well-drained soils with plenty of organic matter, which will provide the best medium for optimum plant growth.
Well, that's it. I hope you enjoyed this video. If you did, please like it on my YouTube channel. And if you really want to hear more, then subscribe to my channel and you'll be notified about upcoming videos, including part 2 of this mineral nutrition of plants series. Have a great day.